So what? Okay. 'Di ba talagang kinakarir ni Mr. Moderator ang kanyang work. I love it. All right. You see it? All right. So next topic that we're going to discuss is the gram-positive organisms. And the important thing that you need to remember is the gram-positive bacilli. Rather, I would like to change this one to gram-positive bacilli because I was intending to write the gram-positive organisms, but then I realized that you have to have gram-positive cocci as a separate discussion itself. So let's try and discuss the gram-positive bacilli. Bacilli. All right. So we're going to talk primarily about the spore formers. Okay. Um, Clostridium and Bacillus species. All right. And then one of the things that you need to remember about them is that you can differentiate them with catalase test. Okay. So for Bacillus species, they will test positive with catalase, and Clostridium will test negative for catalase. All right. So first up, the catalase positive one, Bacillus species. Okay. Now all species of Bacillus are motile except for Bacillus anthracis or the anthrax bacillus. Another term for Bacillus anthracis is the anthrax bacillus. Okay, it is the causative agent of several types of diseases, but I just sorted it out for you. I just sorted it out for you guys. Pulmonary type, gastrointestinal anthrax, and cutaneous anthrax. The one that you need to remember is the pulmonary type because it's known also as wool sorter's disease. All right, sorter's disease is a term that is used for the pulmonary anthrax type of the disease. Okay. Now, the chains of the Bacillus anthracis resembles a bamboo pole or a boxcar appearance when it's observed in the microscope using when it's stained by gram stain. And the location of the spore is centrally located. So I know it's easy for you guys to remember this. I know it sounds simple for you guys to remember this, but just for those who are visual learners, imagine this is the facility. It looks like a pickle. I'm sorry, but for the purpose of the discussion, I'm trying to show off my artistic skills, and this is the extent of my artistic skills. So the spores is centrally located. Okay, as opposed to Clostridium, which is terminally located. This one looks like a poop drawing. So terminally located spores. It could be subterminal in the case of Clostridium tetani, but that will be a discussion all itself on its own by itself. All right. Now we have to talk about the pathologic determinants of Bacillus anthracis. It has two things. You have to remember two things. The capsule is another pathologic determinant. Again, does it cause meningitis? Let's go back. No. So it's one of the few bacteria that has a capsule, but it will not be able to cause. It's rarely associated with meningitis. All right. The most important thing that you need to remember is the anthrax toxin because this is used for bioterrorism. Okay. So they will ask you a question: which of the following microorganisms produces the agent for bioterrorism in the year 2000? I think it was in the year 2000 when the USA tried to invade Iraq. So it was it caused pretty much a lot of scare. It caused a scare to the whole world back then because this one can actually kill you in a matter of minutes. All right. Now let's talk about the staining techniques employed in the demonstration of Bacillus anthracis. You can use gram staining, of course, to demonstrate the boxcar appearance. You can also use Macfadyen stains and immunofluorescent stains if you're staining it for immunology studies. But the most common one in the laboratory is, of course, gram staining, boxcar appearance. All right. Now, in many of the exams that I've taken, even up till now, I'm practicing the profession. I always come across the characteristic colonies produced by Bacillus anthracis, and it is associated with the term medusa head colonies, or in some books, it's beaten egg consistency. All right, beaten egg consistency on blood agar plates, and the consistency of the colonies is in MHA with penicillin is going to appear like string of pearls. All right, so MHA, Muller Hintern agar. All right, so when we're doing what they call this susceptibility testing. All right, now let's move on to the differentiation of Bacillus anthracis specimen that you need to remember. For patient samples, you'll get the encapsulated form. From the culture media, you will see the bamboo form arrangement. All right, so hello. All right, sorry. All right, let's continue. So there is a difference when you look at them directly from the culture media. So for example, you've isolated it and you stained it with the gram stain, and then usually when we get it from patient sample, we have to inoculate first and then make a gram stain. So there is a difference from the clinical sample and laboratory sample. So please be mindful of the appearances. In the gram stain of a patient sample, it will be seen as an encapsulated organism. In culture media, you will see them as the bamboo pole arrangement. Okay, now let's talk about plet. What is plet? Plet is basically a bunch of what do you call this, chemicals and antibiotics to inhibit to be selective for Bacillus anthracis. Polymyxin, lysosome, ethylene diamine tetraacetate, or EDTA, is used to grow your Bacillus anthracis, and it will have the beaten egg white appearance. Okay, that I've described earlier. So Medusa head colonies is in the BAP. Medusa head colonies on BAP, or beaten egg consistency in BAP. All right, and in plet, it will have a beaten egg white appearance. All right. Okay. Now, in gelatin medium, um, the appearance of Bacillus anthracis will grow as what you call an inverted fear tree. Inverted fear tree. What do you mean by inverted fear tree? It would look like this. Okay. So it's like an inverted fear tree. Hello? Okay. Okay. So kung kailan tayo matatapos, saka sila nag-ano? Saka nagkaka...
Sir, medyo nawawala po yung audio niyo po kanina. Nagpuputol-putol po.
Hello? All right. So we're back. kung kailan kung kailan matatapos, saka na ganito yung ano no, yung microphone ko. So anyway, that's what it looks like. It's like an inverted fear tree. Okay. All right. So that's what it looks like. We need to test the if we want to test Bacillus anthracis from animal carcass. Hello, I am. So if we want to test the organism from animal carcasses, you can do so with what you call the S. coli test or the As. coli test. The positive result will be like a precipitation at the point of contact. Now, why do they do this? Because sometimes they test anthrax bacillus for bioterror for bioweapons in animals, and As. coli test is one way to determine if it's actually if it's actually the one that killed the animal, not the impact. So As. coli test is one of those tests. An interesting thing if you're talking if you're using microorganisms for bioweaponry. All right. Now we also have bacillus species associated with food poisoning. This is known as Bacillus cereus. All right, Bacillus cereus, like cereal. The spelling is cereal. Just change the A to U. All right. So there's two types of food poisonings. Okay, that you can get from this disease from this organism. You have the emetic type and the diarrheal type. So, emetic type is because of preformed toxins on fried rice, whilst the diarrheal type is from sauces and meats. See? So, sauces and meats that have been stored for a very long time can have Bacillus cereus in them. Specifically, detox. How would they survive in fried rice, by the way, sir? Remember that these organisms are capable of producing spores, and therefore when there is adverse reaction or adverse effects in the environment, or what do you call this, uh, um, unfavorable conditions in the environment, they would form spores, and they would lay dormant. And as the rice cools down and reach 37 degrees Celsius, it will return back to its original state, or it will divide. It will transform back into its original state and produce toxins. That's why it's called the fried rice bacillus. All right. Now we can differentiate. Um, we can differentiate the Bacillus species using four tests: catalase, lecithinase, motility, and hemolysis on blood agar. All right. Bacillus anthracis will test positive. Bacillus cereus will test positive for all of the tests. Please compartmentalize what you need to learn. Hemolysis would be beta hemolytic. All right. All of the tests will be positive for Bacillus anthracis except for motility. I mentioned earlier, that's the reason why I mentioned earlier that the only non-motile Bacillus species is Bacillus anthracis. And you need to remember also the hemolytic pattern when you're trying to differentiate them with Bacillus cereus. All right. Now, for penicillin susceptibility, we can also use this to identify Bacillus cereus and Bacillus anthracis. Bacillus anthracis is generally sensitive, and Bacillus anthracis is hello. All right, okay, cool beans. Hopefully you guys have absorbed that, because I think uh, we're gonna move on with the contaminant Bacillus species, which is known as Bacillus subtilis. Bacillus subtilis is known as the hay bacillus. Hey, all right, hay bacillus, and the drugs that we use to differentiate them, but the drugs there are certain drugs that are derived from Bacillus species. Actually, bacitracin is one of them, and I think this is a nice to know question. It's from Bacillus licheniformis, and polymyxin or polymyxins, the class of drugs classified under polymyxins, is from Bacillus polymyxia. All right. So please don't forget them. They might be asking the board exam. They might not be asked. All right. Now, next, Bacillus species used as a control organism for mosquito larvae in water system is another one, Bacillus thuringiensis. Thuringiensis. All right. See the usefulness of Bacillus. It can be used as a source of bacteria, antibiotics, as well as control organisms for water systems. Okay. And one of the reasons how they will do this is that they use the spores of this organism. If the spores remain dormant, it means that mosquito larvae can survive. Okay? If the spores become if the spores become active, multiply replicative rods, that means it's not suitable for mosquito larvae growth. And that's one of the ways microbiologists and entomologists are capable working together. All right. Another one that you need to remember, the other Bacillus species for biochemic bio what do you call this, biological indicators. All right, Bacillus Stereothermophilus for your autoclaves, and then there's other ones for the oven and UV system. So please be mindful of them and don't forget them. Okay. Next, we're going to move on with Clostridium. There are two toxicogenic species of Clostridium. Please don't forget Clostridium tetani and Clostridium botulinum or botulinum. Sorry. Okay. So the unique characteristics of Clostridium perfringens is that it is non-motile sucrose fermenter and exhibits double zone of hemolysis and it's capsulated. All right. These are the things that you need to remember about Clostridium perfringens. And then Clostridium species with swollen sporangia. Sporangia na naman, sir. Akala ko ba sa ano lang yan? Akala ko ba sa mycology lang yan? No, they have sporangia as well. But the purpose of the spores for bacteria, as I mentioned before, is for survival. But for fungi, the sporangia is used for reproduction. So see the difference? Okay. So Clostridium species without swollen sporangia is your Clostridium perfringens and Clostridium bifermentans. All right. And Clostridium species that can ferment lactose, Clostridium septicum, and Clostridium perfringens. All right. And organisms under the Clostridium family that cannot ferment glucose, you have Clostridium histolyticum and Clostridium tetani. All right. Next is your only Clostridium that cannot ferment any sugar. So we have, what are the ones that we have? Glucose, lactose, and those are the things that, yeah. So any of these sugars cannot be fermented by Clostridium tetani. All right. There is one aerobic species of Clostridium, that is Clostridium histolyticum. So please don't forget these general characteristics. Now let's talk about Clostridium tetani in general. Other terms that are we have we have the tack head, we have the call this we have the tack head bacillus, we have the lollipop bacillus, the tennis racket bacillus. Call them as you may, whatever you want, but these are the terms that are useful for you. Okay. Now, Clostridium tetani has what you call a terminal spore. As I mentioned before in our discussion of spore locations, Bacillus has a centrally located spore, while the members of Clostridium have terminal or subterminal spores. Now, we have two types of toxins arising from this particular organism. We have tetanolysin and tetanospasmin. You have to be familiar with them because they might confuse you in the board exams. All right. So tetanolysin is responsible for the hemolytic activity of Clostridium tetani, while tetanus plasmin is the neurotoxin that you will encounter when somebody has tetanus or tetano in Tagalog. All right. We also have to note the mode of transmission of Clostridium tetani, and it is through wound punctures. Okay, entry through wound punctures. The unique ability of Clostridium tetani is based on its ability to cause a disease. What is the unique ability? The disease that it causes, or tetanus, is is unique based on the fact that it is capable of causing a disease without actually leaving the site of infection, the portal of entry. How it causes a disease is it releases toxins. The toxins are the one that is responsible for disease, the pathology, pathogenesis of the disease, of what the condition is going to happen. All right. Now we have to talk about what is immediately done to the wound to avoid tetanus. We need to do anti-tetanus toxin administration, of course, within 24 hours, so that we want to prevent or tetanus toxoid. You can use anti-tetanus toxoid. Okay. And as we mentioned before, you have hemolytic hemolysins that is released by Clostridium tetani. And the important thing that you need to remember is that there is a characteristic odor. There is a characteristic odor that is produced by the organism, and that is burnt flesh. So amoy sunog na karne. All right. So please don't forget that. Okay. Now we have to also remember the pathognomonic signs of tetanus. We have trismus and uh, rhizus sardonicus. So these terms translated into English is known as trismus, lockjaw. Rhizus sardonicus is the peculiar smile or devil's grip. Okay, rhizus sardonicus. So peculiar smile. So lockjaw and trismus is characteristic of tetany or tetanus. Okay. Now, Clostridium botulinum is also known as the canned goods bacilli. Okay. There are two types of botulism. One is the classic food or wound botulism because of the ingestion of the toxins. And we have what you call infantile botulism, which is which is transmitted by the ingestion of the organisms or the spores themselves. All right. So infantile botulism, ingestion of the spores, direct ingestion of the spores. Classical slash food botulism slash wound botulism is through the ingestion of the toxins. Okay, or entry of the toxin in wound areas, basically. All right. So what is the classical sign of the characteristic sign of classical botulism or classic botulism? Flaccid paralysis. And then for infantile botulism, we have what you call the floppy baby sign, within which the baby, upon drinking milk that has been contaminated with the spores, they would have like a doll-like appearance. Imagine you're holding a baby, what you call this, a mannequin or a baby mannequin. And that's what a floppy baby looks like. All right. Next, we have Clostridium species associated with pseudomembranous colitis. Please don't forget this. This is one of the most important things that you need to remember because it's on the rise, I suppose, in the microbiology community. This is Clostridium difficile. All right. The media of choice for Clostridium difficile is cycloserine, cefoxitin, fructose agar. All right. You can also use direct testing now. You just need to do drops of suspension, of stool suspension, and then double check for Clostridium difficile. It's now pretty easy for us. Okay. Another term for Clostridium perfringens is Bacillus aerogenes capsulatus, which is basically the causative agent of gas gangrene or myonecrosis. And then we also have the pathogenic determinants of Clostridium perfringens, lecithinase, collagenase, and necrotizing toxins. So those are the three things that you need to remember when you're talking about Clostridium perfringens, because this causes gas gangrene. The alpha toxin refers to what pathogenic determinant of Clostridium perfringens? This is known as lecithinase. It breaks down lecithin in the human body. Okay? And then specimens used for the diagnosis of gas gangrene, we could use wound discharge, pus, and sometimes if if the if the hospital if the patient has undergone surgery, we can use tissue samples. All right? Clat Characteristic hemolytic pattern of Clostridium perfringens is what you call double zones of hemolysis. All right? Double zones of hemolysis is like your alpha prime, but unlike alpha prime, it will produce a double halo. It will produce no gamma hemolysis, but or rather a line that separates the gamma hemolysis and the beta hemolysis and another beta hemolysis. All right. We have a couple of minutes to spare, and also I will have to ask an extension, hopefully from you guys, because we will do the final act of our. Is it okay for you guys? I'll just finish the slides, and then we'll do the final act of our competition that has been ongoing for 4 weeks now. 4 weeks, about 3 weeks, I'm not sure, but yeah, 3 weeks pa lang, no? All right, so let's describe the reaction produced by Clostridium perfringens. It will produce what you call a stormy fermentation. Why is it stormy fermentation on on milk medium? Because it will produce what you call cloud colonies, or it will coagulate the agar and it will look like clouds. Double zones of hemolysis is caused by what toxins? We have the complete toxin and the incomplete toxin. You have to talk about the incomplete toxins. Um, Theta toxins, the inner hemolysis is what you're going to see in the complete toxin. So basically, it will lyse the agar. All right, it will lyse the cells in the agar. Outer hemolysis is the incomplete toxins, which is a little bit lighter, and this is actually caused by the alpha toxin or the lecithinase. All right. Next, we have to talk about the enzymatic tests for Clostridium perfringens, lecithinase, and lipase. And there are species of Clostridium that will test positive with lecithinase and lipase. It's actually lecithinase that would test positive for Clostridium perfringens and lipase for Clostridium botulinum. Let's talk about sugar fermentation. Sugar fermentation is another way to identify species of the Clostridium family. And the only lactose fermenter is the Clostridium perfringens. Remember, lactose is milk sugar, and the only one that can grow on milk medium is Clostridium perfringens. Okay, with a stormy appearance, and all of them will test negative with lactose. All of them will test positive for glucose, except for Clostridium botulinum. Okay, so cool beans. Let's move on. Clostridium, the demonstration that the alpha toxin is on an egg yolk medium, known as Nagler's phenomenon or Nagler's reaction. This is basically a halo opacity around the colonies. Um, Colonies will produce no pitting on colonies without the antitoxin will have pitting. All right. Now, and uh, we need to talk about also the um, zero groups. Okay, so I think this will be the last topic. Zero groups of Clostridium perfringens. They will have five types, A to E, and A and E will be producing the lecithinase, and the group A is the only one related to gas gangrene. Okay. Yeah, there's one more. Another organism that has been associated in rare cases of gas gangrene that is not Clostridium perfringens. It is Clostridium noivii. Malignancy associated Clostridium is Clostridium septicum that has been associated in breast cancer, colon cancer, and leukemia. And I think that's the end. Yeah, congratulations in surviving that. All right. So unfortunately, we were not able to discuss. We weren't able to discuss uh, spirochetes, mycobacterium, and I think um, Listeria and Erysipelotheryx. So I want you guys to take that as an assignment. So we have only three topics that we haven't discussed. Hopefully, 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 you remember everything that I've taught you. And if you haven't, you can visit the channel, and I've prepared a playlist for you. Now, would like to ask the following students uh, to participate in my last and final exam, final competition. These are the people. That's not the one. Am I sharing it? No, no, no. That's not the one, huh? I prepared a slide for that one. No, that's not a slide, but I can't find it anymore. So I'm gonna stop the presentation. And congratulations in surviving this annoyingly long lecture. And thank you so much. It has been such an honor to teach each and every one of you. One of my favorite subjects is actually microbiology, but it doesn't include virology. Now I have to ask these three students, Adrian, um, Joanna, Baltar, and Mayra. They're the ones who are the top scorers in my ongoing lecture presentation under Kahoot. O, di ba? The highest one will be revealed later. Okay? I will send it to Sir Mac, or I will send it to somebody from the class who has added me on Facebook. Now, unmute yourself and show me yourselves in the camera because I need your help in preparing this. Adrian, Mayra, and Joanna Baltar. You need to have a piece of paper and you need to write A, B, C, D on those pieces of papers. I have to see it. Joanna, Mayra, Adrian. Okay, so this will be a time trial challenge, and each point will be added to your each. There's only 5 questions in my exam, and it's not going to be played on Kahoot because I lost my subscription. So where are those three students that I've asked? Eh, they're not participating. Ay, wala si Joanna. So we have to take the other another person. So we have to talk to the other person. Ay, ayan na si Joanna. Okay. Ayan, oh, di ba? Ready, ready. Lumipat ng ibang kwarto. Gusto ko yan. So we're going